So now we move on to our hot topic presenters. And the first one of those is Professor Ranjeny Thomas. Ranjeny is a Professor of Rheumatology at the University of Queensland Fraser Institute, Translational Research Institute, consultant rheumatologist at Princess Alexandra Hospital and Fellow of the Australian Academy of Health and Medical Sciences, and a member of the Order of Australia. Her research is focused on the study of autoimmune disease and the restoration of tolerance. Ranjeny.
Thank you, Stuart, and thank you very much for having me here today. So I'm going to really try to draw some similarities between rheumatoid arthritis and type 1 diabetes and bring that to some learning for us. These are my disclosures. Okay, so firstly, rheumatoid arthritis is an autoimmune inflammatory disease of the joints and other diseases, different from type 1 diabetes where the median onset is 55 years, but similar in many other ways. We have a specific autoantibody, anti-citrullinated peptide antibody, present in 70% of patients, and we call that ACPA. Patients have intermittent symptoms before disease. There's lifetime treatment with disease-modifying drugs available. But interestingly, there's a window of opportunity when they're most effective, and that's in the first year after onset. And we can get about 70% of patients into remission on combined disease-modifying drugs. But patients tend to lose that remission over time, and part of that, like in type 1 diabetes, is related to compliance. And so for people to stay off drugs and in remission for at least 5 years is quite rare. Now, type 1 diabetes is, again, autoimmune disease of the pancreatic islets and much younger age of onset. Again, autoantibodies precede diagnosis, and there's intermittent hyperglycemia in stage 2 before the onset. We've got no disease-modifying drugs approved after onset, and whereas teplizumab is approved in stage 2. And again, we've got this variable duration of partial remission after, for up to 2 years after onset. And I think that this is really a window of opportunity for type 1 diabetes as well. So in looking at the T cells that were pro-inflammatory in the joint synovial tissue, using transcriptomic analysis and single-cell T-cell receptor analysis, we got a bit of a prize, the group of investigators here led by Pascal Weir, who did this in her PhD. And what she found was that the most clonally expanded and pro-inflammatory cells making interferon gamma and TNF were actually cytotoxic T cells, which hadn't really been thought of as being part of rheumatoid arthritis. And when you look at what they're recognizing, many of them were recognizing CMV, EBV, and common chronic viral infection. And we went on with Maria-Pierre Esposti and her team to do an animal model of mice with latent CMV and then induced antigen-induced arthritis in mice with or without latent CMV, and came up with a model actually in which there's cross-licensing through the cross-presenting dendritic cells. So we've got the dendritic cell here that's cross-presenting both viral antigen and self-antigen, and there's this cross-licensing of CD8 T cells to become more activated and make cytokine, and CD4 self-reactive T cells to be more activated effector cells. And then we've got the pro-inflammatory stromal cells producing things like IL-6, and also we saw in type 1 interferon signature in the stroma. And of course, those T cells help B cells to make autoantibodies, and the follicular helper T cells are very, very relevant to the rheumatoid disease process. Now, in type 1 diabetes, we envisage this to be really quite similar in the islet, and we've got the target of the CTL here, the beta cell. And if you think about the treatments that we're giving in rheumatoid arthritis, and you've been trialling, this is where they operate with the TNF inhibitors, the teplizumab, the JAK inhibitors, and in rheumatoid at least, IL-6 receptor inhibitors, abatacept, and anti-CD20, and abatacept here as well. Now, really important to the further discussion of tolerance is that the phenotype, the signalling, and the location of the CD4 self-antigen specific T cells is critical to the outcome of this interaction. They can be effector cells and they can be regulatory. And it's interesting that we know the autoantigens, at least the ones that matter in both type 1 diabetes and rheumatoid arthritis. So something else is, where do these CD8 CTL come from? And some very important work done by Andrea Scheetinger's lab, and she comes from a cancer background, is looking at the origin of the IGRP-specific CTL in the NOD mouse model. And she found in the pancreatic lymph node that there were two populations, a TCF1 high and a TCF1 low. And this TCF1 high gave rise to an autoimmune stem-like progenitor population that in transfer studies was able to transfer type 1 diabetes. In contrast to TCF1 low had the autoimmune mediator phenotype. However, if they blocked the further movement between the pancreatic lymph node and the pancreas, in fact, type 1 diabetes just petered out because these cells were short-lived and were no longer able to prolong inflammation. So in tolerance, what we're really looking at is in bringing in regulatory T cells to control either here or here through this cross-licensing, cross-tolerance. So where do regulatory cells come from? Well, they're originally born in the thymus, and they come from single positive T cells, and those single positive T cells need to see antigen presented by antigen-presenting cells, and that needs to be long and slow, and then it needs to be interrupted, and that interruption signal was shown by Al Singer's group earlier this year is TGF-beta. The regulatory cells regulate effector cells. Now, the same thing is going on in the periphery. We've got dendritic cells which have low levels of co-stimulation, are presenting low levels of self-antigen, and leading to the development of antigen-specific anergic T cells. When that antigen presentation is interrupted, again by TGF-beta, regulatory T cells develop. And we have specialized systems that have evolved in the gut and the gut-draining lymph node for this to happen for our food and commensal antigens. Importantly, these anergic cells can express CTLA4, and when that interacts with CD80 on the dendritic cell, it can pull the CD80 off the dendritic cell, exposing more PD-L1, the checkpoint molecule. So we developed an antigen-specific immunotherapy for suppression of pathogenic responses while maintaining protective immunity. And we've demonstrated that there's low-level antigenic peptide presentation from a liposome. So the liposome here is about 100 nanometers in diameter. It's got an immunomodulator in the shell, calcitriol, which blocks NF-kappa B in dendritic cells. And then in the core, there's a peptide, and this can be substituted out depending on which disease we're treating. And we showed that the liposomes are taken up by dendritic cells in the draining lymph node. You can see the liposomes arriving here in the draining lymph node and coming to be taken up by the CD11C dendritic cells. And these dendritic cells, it turns out, constitutively express PD-L1, and that they lead to expansion of a PD-1 CTLA4 anergic T cell population that can then become regulatory, and this is PD-L1 dependent. And here is the PD-L1 expression. This is where the liposomes are presented right around the follicle in the lymph node. Now, another important thing about this immunotherapy is cross-tolerance. So we demonstrated this with Emma Hamilton-Williams' group, and here's a liposome that encapsulated the CD4 mimetope, BDC2.5, in the NOD mouse. The regulatory cells were expanded in the draining lymph node, the inguinal. They're able to recognise their antigen in the pancreatic lymph node to modify the dendritic cells there, and so cross-tolerise the IGRP-specific CTL in the pancreatic lymph node, leading to beta cell survival. So we translated this and we developed the drug Gen181 by a spin-off company that we spun out of the University of Queensland in collaboration with Janssen Biotech. And these liposomes entrap a collagen epitope and calcitriol, and the epitope is restricted by 2 of the HLA molecules associated with rheumatoid arthritis. Our primary objectives in this small trial, single ascending dose trial, was safety and tolerability, as well as immunomodulation, looking at the antigen-specific and the bystander T-cell CD4 response, using specific tetramers, and we looked at the clinical response as a secondary outcome. And we had 3 different doses beside the placebo, 0.3, 1, and 3 mils of liposomes, and all patients were on methotrexate monotherapy. So to sum up, because this has been published, it was safe and well tolerated. All patients receiving the 0.3 and the 1 mil dose were in remission by 8 weeks after a single dose. And one patient actually started with active disease who received the highest dose, flared over the first 2 weeks. So we were really interested to see whether we could find from our biomarker panel markers, phenotypic markers that were associated with improvement in the disease activity score in patients who received the drugs, and we did this by k-means clustering. And what we found was that an expansion in the number of collagen-specific or Vimentin-specific T cells was a good outcome, and the phenotype more naive and more CCR7 positive, but not good if you were expanding effector cells or had inflammatory dendritic cells or monocytes. So we've developed a concept of applying a tolerizing antigen-specific immunotherapy to prolong remission in autoimmune disease. And so our experience with the high dose, it's kind of to be expected that we know we need low doses and low co-stimulation intolerance. So we think that it would be best to achieve remission or low disease activity in patients who have got new onset disease on disease-modifying drugs, and then to give them antigen-specific immunotherapy to try to extend that remission. We know that they lose it by year 2 often, and then to try to drop the DMARDs and monitor them with specific biomarkers, and we're starting to get an idea of what those are. So we recently looked at, in the peripheral blood, citrullinated Vimentin-specific T cells in patients in our clinic with DMARD remission, and those who've got active disease. And remarkably, we found that in remission, there was an expansion of naive-looking citrullinated Vimentin-specific absolute number of T cells that lack inflammatory site homing receptors, and in active disease, they were contracted, just like in our trial, and they had the chemokine receptors for entering into inflammatory sites. So again, in type 1 diabetes, we're first targeting a trial after onset of disease, and our aim is to in the future, in efficacy studies in the future, to extend the duration of partial remission with antigen-specific immunotherapy. So what that means, we need to find T-cell biomarkers that predict remission and use those to monitor disease. So Yasmin Mustafa used a clinical index that Kerry Buchanan had developed during her PhD, and Yasmin used the CFSE assay for proinsulin that Michelle So had published from Stuart Mannering's lab, and found that the higher the level of proliferation towards C-peptide, the longer the remission, and here you can see the cutoff of greater than 3, had a much longer duration of partial remission. And when Yasmin went on to look at what was the phenotype of these proliferating T cells, she found that in the CCR6 negative population, that the lower the CXCR3 and the higher the CCR4, the longer the duration of partial remission. And this was in blood taken 3 months after onset. So our model is like this, that we've got proinsulin-specific T cells that express CXCR3, and have the propensity to go towards their chemokine ligands in an inflammatory situation in the islet, whereas those that are CCR4 positive go towards the tolerogenic dendritic cells in the lymph node, and this keeps them regulatory, can regulate in a bystander manner, but also keeps them out of the islet. So Gaia M in our group has also been looking at fresh blood, and she's also found that high CCR4 amongst the total CD4 T cells and low CXCR5 in the C-peptide-specific T cells also predict long remission. So the same idea that the low levels of the chemokine receptor to get them into the follicles where they could help B cells. So we are very excited that we've just started the tox batch for our liposome immunotherapy trial, which will be undertaken in the first half of next year. It's a liposome that entraps the C-peptide and calcitriol, and we're going to be doing a phase 1 study next year for safety, looking at the number and the phenotype of the antigen-specific T cells and the bystanders, and looking at beta cell function, glycemic control, and IDA antibodies. Just remains to thank the many people that have been involved in this work, particularly those that developed DEN181 and continue to work on immunotherapy development in type 1 diabetes, and the nice collaboration that we've had with Emma's group and with my students that have done that lovely biomarker work, and our commercialisation team and the funding bodies. Thank you very much.
Thank you very much, Ranjeny. Our next speaker is Dr. Irina Buckle. Dr. Buckle is an immunologist in Josephine Forbes Laboratory at the Mater Research Institute in Brisbane. She works on the development of disease-modifying therapies for type 1 diabetes, specifically focusing on the receptor for advanced glycation end products, RAGE. Irina.
Thank you very much, Stuart, and I'd like to thank the Rota and the organisers for this incredible opportunity as an early career researcher. You can imagine standing on this stage is both terrifying, particularly speaking directly after and directly before one of the both incredible Thomas sisters, so bear with me. But I'm very excited today to show you some of our work, that our piece of jigsaw puzzle that we are hoping to contribute to that very incredibly complex type 1 diabetes puzzle. And I did look at the program, and I knew that everyone will cover the introductory slides very well, hence I do not have a slide to what type 1 diabetes is, and I'm going to dive straight into a receptor for advanced glycation end products, or RAGE. And essentially it is immunoglobulin receptor, superfamily receptor, and the key message here that essentially binds a lot of ligands, and insulted, infected or injured pancreas contributes to some of that ligand release, and what happens downstream is important. It leads to inflammation, and inflammatory process perpetuates and results in further damage and reactive oxygen species production, and so on and so forth. And the soluble RAGE is a truncated isoform that can act as a decoy receptor, binding those ligands and preventing the downstream signaling. And as I mentioned, there are a number of ways of preventing this from happening, either preventing the ligand receptor binding interaction or indeed the downstream signaling. We've been, our team rather, has been heavily involved in some of the work, and this work has also been pioneered by Professor Harold and his team a number of years ago, where it has been described that RAGE increased in islet antibody positive individuals, and it is associated with progression to type 1 diabetes. Professor Forbes, in collaboration with Professor Mikhail Nib, showed that RAGE polymorphisms are predictive of type 1, and the current work that is we are hoping to share with type 1 diabetes community very shortly, led by very talented Sherman Lung in Josephine's lab, in collaboration with Professor Pugliese in NPOD, also talks about human islets and increase of RAGE in the NPOD donors. And of course, lower soluble RAGE is also associated with more severe type 1 diabetes. And that is, again, in collaboration with the Findayan study and Professor Mikkel Nib's DIP study. So what I'm going to talk to you today is the ways of stopping this from happening, and by stopping this from happening, I mean RAGE antagonism and arrest of type 1 autoimmune-like disease development in preclinical mouse models, and increase of functional Tregs and increase in pancreatic islet health. So that's our way of raging against type 1 diabetes. We have a number of therapies in the pipeline, some of which I'm not discussing today, but essentially we have some novel approaches delivering soluble RAGE, repurposing oral RAGE antagonist drug that already has been tested in humans with reasonable success, and other RAGE therapies. So another interesting point about RAGE is that its gene, AGR, is located in MHC class 3 region, known of genetic susceptibility to type 1. And this very complex figure, the take-home message from this is essentially it's a very good data set published a number of years ago. This group was dendritic cell biologists, but what it says that essentially RAGE is expressed on lots of immune cells at a way above detection threshold, and some of those plasmacytoid dendritic cells and the rare population of cross-presenting CD141 positive disease. There somewhere, sorry, I can't see, but they are indeed expressing RAGE, and it is a very exciting data set, and that we are hopeful to explore a little bit further in the very not so distant future. This is a preliminary data set from young people in Matach Young Adult Health Center. These are 16 to 25-year-olds, young individuals with type 1 diabetes duration less than 2 years. And essentially what you are looking at is a surface RAGE expression on CD3 positive T cells and CD3 negative non-T cells, and you can appreciate this flow cytometry panel is not super comprehensive. So when I mean CD3 negative non-T cells, it's everything, including dendritic cells and myeloid cells, etc. But essentially, it's almost a reverse relationship. So RAGE surface expression is increased on T cells, and well, almost a reverse relationship with let's call it myeloid compartment. And this is the work that I alluded to in my original slide that is currently is in review, led by Sherman, and you're looking at immunofluorescence imaging with control islet at-risk individuals and type 1 diabetes individuals, and you can appreciate that both RAGE and one of the RAGE ligands, AGEs, are indeed increased in type 1 diabetes patients and individuals with at-risk individuals. And in the interest of time, I'm going to move on into the preclinical mouse study, again led by Sherman, and that's now published in Diabetes, and it is a soluble RAGE work whereby it's been delivered for a short period of time, just the 2 weeks, and you can appreciate that it significantly delays autoimmune diabetes development in these animals after a very short treatment. And it was, of course, a biologic, so now we are going to move on to testing a low orally available RAGE antagonists. And in this study, mice were treated from day 50 of life, about 7 weeks old, NOD mice, and daily for 2 weeks and 3 weeks thereafter until about day 150. And we monitored these animals for progression of autoimmune diabetes. And again, you can appreciate it's a modest delay, but it is a statistically significant one, where our orally treated orally treated mice with RAGE antagonist delay type 1 diabetes, autoimmune diabetes development. We looked at immune profile of the cells just after the 2 weeks of treatment, and for some reason my stars aren't showing, but it is significantly, the Tregs significantly increased in spleen, and the number of proportion of those Tregs are unchanged in pancreatic lymph node. However, when we looked at function of those cells, we see that these Tregs are functional, and there is increase in both ICOS and TIGIT expression in both pancreatic lymph nodes and spleen, not so much TIGIT, but it is the right trajectory. When we look at CD8 positive T cells, interestingly, they are slightly increased. I'm sorry, I'm not sure what happened to my stars, but they are slightly increased in spleen and slightly decreased in pancreatic lymph node. But once again, when we look at their function, there is indication of exhausted phenotype with increase in PD-1 and TIM3 expression levels in pancreatic lymph nodes. When we looked at the beta cell function after the treatment, and that is on the left, you're looking at insulitis scoring, and all of you are familiar that the grade 0, it's no immune cell infiltration, all the way to grade 4 with severe infiltration. These mice have increased or rather reduced immune cell infiltration in treated groups. This acute insulin release graph here is the first phase insulin after the oral glucose tolerance testing after 100 days of treatment. To summarize, we I'm very hopeful that this treatment, we show that this treatment increases functional Tregs and spleens and pancreatic lymph node, and hopefully leads to exhaustion of CD8 positive T cell, hopefully reaching both arms of tolerance. We show some reduction of insulitis in the pancreata of these animals, and improvement in acute insulin release and delay of type 1 diabetes, and of course WER2, and we've got lots of things in the pipeline, and one of which is in-depth characterization of T and antigen-presenting cells following the treatment, and of course with the approval of teplizumab, it is important for us to, although mouse hamster equivalent is not teplizumab, but in preclinical models, that's what we have to do. We are collaborating with Queensland Children's Hospital, and we are hoping to look at risk in newly diagnosed individuals and assess RAGE profile on immune cells in a large flow cytometry panel, and have a more comprehensive overview as to what's happening in peripheral blood of those individuals, and of course testing other RAGE targeting therapeutics for secondary prevention. There are a lot of people to thank, the team who's done most of the work, and Professor Kevin Harold and his team for RAGE biologic side of things, and his collaboration has been very important in this process, and NPOD, families and donors, and Professor Pugliese and our funders, and of course JDRF and our biotech collaborators. And thank you.
Okay, 2 more talks and then you can have a cup of coffee. So our next speaker is Professor Helen Thomas. Helen is head of the Immunology and Diabetes Unit at St Vincent's Institute in Melbourne. Her research is focused on prevention of type 1 diabetes, and her work has been applied to humans through the transplantation of islets isolated from organ donors by the Tom Mandel Islet Transplant Program, and she has also developed collaborations with industry to test immune suppressive drugs, in particular JAK inhibitors, in clinical trials for type 1 diabetes. Helen.
Thanks, Stu. Thank you for giving me the opportunity to present today, and I really want to thank the JDRF for letting me and Tom both showcase the effects of JAK inhibitors in type 1 diabetes. It's a real pleasure to do that. So I'm hoping that everyone in this room saw Tom's talk, because I'm not going to give any of his, well, I'm not going to go through any of the stuff that he presented. But what I'm going to do today is tell you about some of the preclinical work we did looking at the mechanistic basis of the use of JAK inhibitors, and some of the data that we showed to the drug companies that made them enthusiastic about using their drugs in our trial. So as Tom showed you, with a JAK inhibitor, baricitinib, we can stop the progressive loss of insulin production in people with type 1 diabetes, and that's this data again showing on the left. We also know that we can stop the progressive loss of beta cells in NOD mice, and that was obviously done before the human study, and that's shown on the right there. So I'm not sure if you can see, but in this study we waited for mice to become diabetic and then treated them with a JAK inhibitor from AbbVie called ABT317 for 40 days. There's supposed to be a box around here that shows when we treated the mice. And over that 40-day period, the mice remain normoglycemic when they're being treated with the drug and not the vehicle-treated mice. This one. Now, having seen that result, I'm going to go back and give you some of the background to how we got to studying JAK inhibitors in diabetes. So we know that CD8 T cells interact with beta cells through their T cell receptor and on the T cell and the peptide MHC presented by the beta cell. And we did some videos, actually Prerak is in the audience today, he made these videos during his PhD, where we took a beta cell, which is this big cell in the middle of the field, and we imaged the T cells forming an immunological synapse and killing those beta cells. So I'll start the movie and then I'll explain. We've added a calcium fluorophore that goes green when the T cell forms a synapse with the beta cell. And you'll see that quite quickly here. And then we've also added a dead cell marker, propytimidide, so we can see that the T cell kills the beta cell, and then the T cell actually leaves the beta cell, goes off to kill another beta cell in the culture. So this is really nice sort of graphic evidence of the beta cells being killed by the CD8 positive T cells. And we've also known for quite a long time that targeting the JAK-STAT pathway can prevent diabetes in NOD mice, and I've just got two studies here on the left. First one with STAT1 deficient NOD mice. So JAKs are direct activators of the STAT proteins, which are downstream of cytokines, and these STAT1 knockout mice are protected from diabetes. And then on the bottom there is our studies using overexpression of the suppressor of cytokine signalling, or SOX1, in the beta cells. And SOX1 is a negative regulator of the JAK-STAT pathway, so if we overexpress it, then we can block JAK-STAT signalling. And at the same time that we were doing these studies in mice using knockouts in transgenic mice, the pharmaceutical companies were developing inhibitors of the JAK-STAT pathway. And these inhibitors bind to the ATP binding pocket of the JAK molecule, shown here, and inhibit the kinase activity of the JAKs in a way that's very similar to how SOX1, which is this molecule here, inhibit the JAK-STAT pathway. So the JAK-STAT pathway is downstream of many cytokine receptors, and I'm going to just talk you through a couple of those different families and some of the data that we have. So first of all, looking at the interferons. We've known for quite some time now that the upregulation of MHC class 1 is a hallmark of type 1 diabetes. On the left here is some data from Sarah Richardson's paper showing an increase in increase in HLA staining in the islets of organ donors with type 1 diabetes, and also an increase in STAT1, which is the signalling molecule that I described earlier. Class 1 can be upregulated on islets by culture with interferon gamma, and that's, that's shown here, although I can't see the line. There's a line here. You probably can't see that either, showing an upregulation of class 1 by interferon gamma. and we can prevent that when we use a JAK inhibitor. So some work we did a long time ago using our SOX1 transgenic islets, we showed that if we block the ability of beta cells to upregulate class 1, we can block the ability of CD8 T cells to kill those cells. And that's shown in these 3 bits of data. So we've got, over here, we've got the SOX1 transgenic islets in green, and when we use an effector to target ratio in a cytotoxicity assay of 10:1, we can see that those islets are protected from being able to be killed by beta cell specific CD8 T cells. And in our transgenic SOX1 transgenic mice, we see the upregulation of class 1 MHC that we normally see on beta cells in wild-type mice is completely prevented in these SOX1 transgenic mice. And these mice are protected from CD8-dependent diabetes. So we then tested to see whether JAK inhibitors that act in a similar way, as I said, to SOX1 would have the same effect. So here we've treated our NOD mice at a stage before they develop diabetes, but after they've developed considerable autoimmunity. We've treated them for 2 weeks with a JAK inhibitor and just looked at class 1 upregulation. We can see this is the level of class 1 on beta cells in our vehicle-treated mice, and here's the level in the JAK inhibitor-treated mice, which is remains at baseline levels. So then going back to the videos that Prerak made, and here we've used beta cells again, but this time we've either added a vehicle to the culture or we've added the JAK inhibitor to the culture. So if I start this, the top one is the vehicle-treated cells, and you can see again the very bright green calcium flash that occurs. And then if I start the bottom video, this is the one that has the JAK inhibitor. Here we can see the T cells are sort of swimming around the beta cell. They're not really forming very long synapses with the beta cell. And in the data here, you can see that the calcium flux is reduced, the synapse time is reduced, and the ability of those T cells to kill the beta cells is also reduced. So you might say, oh, so the mechanism of JAK inhibitors working is them blocking interferon gamma signaling in the beta cell. Well, we know that if we knock out interferon gamma receptors in NOD mice, we can't prevent diabetes. And that's just shown in some data we produced recently here. And I could give another talk about why we think that's the case, but I'm not going to do that today. But what I am going to do is just talk about a different family of cytokines that's also signals through the JAK-STAT pathway, and that's the common gamma chain cytokines. And these cytokines use the common gamma chain of the IL-2 receptor, and they're important for the homeostasis and proliferation and activation of T cells. We know that when we culture T cells with JAK inhibitors, we can prevent the expansion of T cells. So there's just 2 studies here, one in vitro showing if we culture islets that have been isolated from NOD mice with IL-2, we get an expansion of islet antigen-specific CD8s that recognise the antigen IGRP. And if we use a JAK inhibitor, we can prevent that expansion. And then if we treat NOD mice in vivo with a JAK inhibitor, we can also see a reduction in the proportion of cells that are proliferating, so they're expressing BRDU, compared to the vehicle-treated mice. So we can block the expansion of these cells using JAK inhibitors. And we can also see a reduction in the effector CD8s with JAK inhibitor treatment. So here again, we've treated them at this time point just before the development of diabetes. And we see here a reduction in this proportion of cells that are antigen-experienced effector memory T cells. And just looking at our data from the BANDIT cohort, we also see a reduction in these equivalent population of cells in those people on baricitinib. And so all of this effect of JAK inhibitors on T cells and on beta cells translates to a reduced insulitis and diabetes. So here we've got the pancreas sections of mice that are treated with JAK inhibitors compared with vehicle. We can see a lot more of the islets don't have any infiltration. They have a score of 0, and there's much reduced insulitis there. And in this data that I showed you before, the mice are protected from developing hyperglycemia so long as they're treated with JAK inhibitor. And this is quite different from our SOX1 transgenic mice, where in those mice we're really just protecting the beta cell from the effects of JAK-STAT cytokines, not the T cells. And you can see the insulitis there. The beta cells are protected, but they're surrounded by a very big inflammatory infiltrate in the pancreas. And so even though we can prevent diabetes, we've got this big infiltration of T cells. So we've got sort of quite a distinct phenotype despite the same effect on diabetes. And so this is a summary of all the stuff that I've shown you just now. So JAK inhibitors can block the ability of the CD8 T cell to recognise and interact with the beta cell, and then the activation of those CD8 T cells. So it works on the beta cell, but it also works on the common gamma chain cytokines to block the expansion of CD8 T cells. And so I guess if you go back to the diagram that Colin showed in his talk, showing the many different places where immunotherapies have been tested, antigen presentation, cytokines, T cell activation, a JAK inhibitor fits into all of those categories. So I think you can put it into all the different boxes. And then I promised Sanjoy that I would finish with one slide of new data. And so we're interested in trying to develop therapies that have a short-term treatment with a long-term benefit. And what you can see in our mice here is that when we stop the JAK inhibitor treatment at 40 days, eventually these mice do develop diabetes, which I guess is what you might expect as long as they're being treated with the drug, which has a relatively short half-life. Then it's acting on the cells, but once you take that away, then the cells are able to repopulate and continue to cause beta cell destruction. So we've had a go at combining the JAK inhibitors with anti-CD3, and what we reason is that these are targeting two different pathways. The anti-CD3 is targeting the T cell receptor, and the JAK inhibitors are targeting the cytokines downstream. And so this diagram's not quite right, but what we are suggesting is that with the anti-CD3 treatment, we are targeting the T cell receptor and making those T cells dependent on the on cytokines for proliferation. And if we then block their dependence on cytokines by using a JAK inhibitor, we might be able to remove those cells or inactivate them permanently. And in some very new data that's just not actually even completed, we've got these NOD mice that we've treated with anti-CD3 and JAK inhibitor over this 2-week period here. We can see the combination therapy is in pink here, and we get, it looks like a nice protection from diabetes. I don't have any p-values on here yet because it's still ongoing, this study. And with that, I'd just like to thank everybody, especially Prerak, Ting Ting, who did most of this JAK inhibitor work. And Prerak actually is back in our lab and he's doing the combination therapy work too. And then, of course, all of the people involved in the BANDIT study, many previous lab members who did a lot of the older work that I showed. A team of research assistants, both past and present, who've done a lot of the work here. And the collaborations that we've had with people in industry who've given us their JAK inhibitors for testing, they've really been sort of helping us to champion this project. And also the funders, thank you very much.
Thank you very much, Helen. So our final presenter for this session is Dr. Andrew Sutherland. Andrew is a Senior Research Officer in the Immunology of Diabetes Unit at St Vincent's Institute. His primary focus lies in the understanding of the regulatory mechanisms governing the activities of effector and regulatory T cells in type 1 diabetes. Andrew.
Great, thank you, Stu. Let me just get my bearings here. All right, so we've seen various versions of this already today, so I'm not going to spend much time on it other than to just really reiterate a couple of key points, which is, you know, if we can identify what the key affected pathways that are involved in the autoimmune response are in type 1 diabetes, these are probably going to be some of the best chances for immune intervention, as well as trying to identify pathways that are able to maintain or enable the Tregs to function more effectively throughout the entire process. And that's an ongoing, excuse me, that's an ongoing interest of ours in the lab, and particularly in response to the data that's come out recently with anti-CD3 therapy. It's really refocused our attention to on the T cell itself in the pathogenesis of type 1 diabetes, and has again suggested that we need to continue down this path and really continue to understand at a deeper level what the important parts of the T cell response are. So over many years we've come to learn that there are these subsets of T cells which have evolved various roles within the immune system and are characterised by different effector lineage, transcription factor patterns, and also cytokine production patterns. Now, to date in type 1 diabetes, we'd have to say most of the research has sort of been along this arm of the sort of interferon Th1 type T cell, but there's now evidence that there's multiple other sorts of affected lineages out there that may also be contributing. So more recently there's been a lot of data coming out around IL-21 producing cells, here denominated by the TFH lineage, and also the important role of the Treg as a suppressive type T cell that keeps all of this under control. We've focused our attention over the last decade or so, particularly on this particular subset here, the Th17 type T cell, which exemplify or have particular characteristics that separate them from the other lineages. They are around the sort of cytokines that induce the response, and they're listed here. A particular note, things like IL-23, a particular transcriptional program that is mediated by a couple of different transcription factors denominated here, raw gamma T and raw alpha. Expression of this important receptor, IL-23 receptor, which leads to the terminal differentiation of these cells, and a variety of different output cytokines that are critical for the function of these cells. And I'm going to focus today particularly on a couple of different IL-17 family members. All right, so I've just been through that schema. At the initiation of these projects, probably 10 years ago, there was a few different pieces of information within the mouse literature in particular that indicated that these pathways may be important in the development of type 1 diabetes. This is some data of ours showing that if you generate a NOD mouse that's deficient in the IL-23 receptor, you see this significant protection from disease. Another group out of the US showed here that with this particular raw gamma T, raw alpha, dual inversed agonist in NOD mice, you can prevent the development of type 1 diabetes in the NOD mouse, suggesting that those transcription factors are also important in the developing response. And in addition, a little bit of data, slightly conflicting data, suggesting that while a couple of different knockout mice for IL-17 are not protected from diabetes, treatment of NOD mice in a preventative regimen with anti-IL-17A antibodies was able to achieve this protective effect here. In addition, there were a couple of smaller clinical studies that indicated that here in a couple of cohorts of children with new onset type 1 diabetes, that there was indications of increased levels of Th17 associated cytokines and transcription factors, suggesting that it was correlated with the diabetes immune response in some way. Now, why is this important? There's been a huge amount of clinical development in this area over the last couple of decades in terms of mapping the molecules within the different pathways and then developing specific inhibitors to these pathways. And I think this gives us some increased opportunity for developing new strategies in clinical trials, incorporating these different molecules. However, there's I would say, a general lack of mechanistic understanding underpinning a lot of this work within the type 1 diabetes, in that we probably don't know as well as we need to how all of these things function together. And of course, not knowing how the mechanisms are operating at a precise level, it's not clear which candidates would be best to move forward with. All right, so we have a number of projects in the lab where we're looking at a variety of these different points of intervention, whether it be at the level of, the cytokine outputs here or transcription factors, etc. I'm just going to show you some data today around our work on the IL-17 cytokines and receptors. So, I'd have to say that most of the work in this field around IL-17 cytokines is focused on IL-17A, and that signals via this receptor complex here, IL-17RA-RC heterodimer. But it's important to note that there's a closely related cytokine, IL-17F, which also signals through this receptor and the capacity for a heterodimer to also form. So there's a level of complexity here that hadn't been addressed in type 1 diabetes. Probably the most accepted model of the function of these IL-17 cytokines, or IL-17A in type 1 diabetes, was IL-17 producing T cells existing within the inflamed islet and mediating their effects both to other immune cells within the islet, but also directly towards beta cells and potentially inducing inflammation and death within those particular cells. So that's the model that we sort of set about trying to understand IL-17F in the context of. The first thing we did was to look into the islets of NOD mice and demonstrated that there were detectable populations of IL-17F producing cells here on the y-axis, both IL-17A and F double producers and single producers, some evidence of a dual population here of IL-17F interferon gamma double producers in the islets, and they were IL-10 negative, suggesting that they were likely to be effector type Th17s. And I've just shown that graphically here. So we then did some further work to understand how the signalling would be operating within those particular cells, within the beta cells. The first thing we did was to map the receptor expression with islets, and showed that in a panel of a couple of different beta cell lines and islets, that there was a strong expression of both IL-17RA and RC, with this additional expression of RD, a less well-characterised receptor. When we then stimulated a couple of different beta cell lines and primary node islets with both IL-17A and F, and measured the expression of a very acutely expressed transcription product, this NF-kappa-BIZ molecule, we showed that IL-17F was able to induce a response, an acute response, similar to IL-17A in this context. Excuse me. We then did a little bit of work with some CRISPR knockouts and showed that in cell lines that we've knocked those two receptors out, they lose the ability to respond to both IL-17A and F. We then moved on to look at some pathogenic effects of IL-17F in some isolated mouse islet cultures. The first thing we did was recapitulate that data from the cell lines in this particular transcript, NF-kappa-BIZ, and showed that both in isolation here, IL-17F and IL-17A induced this transcript, and also in this second half of the panel, in a cocktail of cytokines, including TNF and interferon gamma, which has been shown to be an important milieu for enhancing the function of IL-17 cytokines, that we saw similar effects. Interestingly, within the context of this cytokine milieu, we saw the potent induction of inflammatory factors, including chemokines here, in response to IL-17F. And when we studied a panel of genes associated with beta cell identity and function, we could see that when IL-17F was stimulated those cells in the context of the inflammatory cytokine panel, we saw this potent downregulation of a whole bunch of transcripts associated with beta cell function. When we then did some functional assays, we were able to show that IL-17F was able to prevent glucose-stimulated insulin secretion, and in addition, that stimulation with IL-17F led to increased levels of apoptosis in primary islets, both from NOD and B6. So the summary from that part of the talk was essentially that we feel like we've identified some sort of very similar functions of IL-17F compared to IL-17A in the context of islet function and islet inflammation. We then wanted to test what the in vivo relevance of that would be, and we took the approach to create genetically modified NOD mice that had knocked out both of those receptor subunits. So to confirm the function or the validity of those knockouts, we stimulated isolated islets from both the IL-17RA and RC knockout mice with these cytokines, and showed that they'd lost, both of those lines had lost the ability to respond to IL-17 cytokines. However, when we did the diabetes incidence studies, we saw a completely divergent phenotype emerging, which doesn't fit clearly with this model that's being presented over here on the left-hand side. When we compare the phenotypes of those two mice, we can see that the IL-17RA knockouts, which are protected from diabetes, have a significantly reduced islet insulitis score compared to the susceptible RC knockouts. And interestingly, both lines had some level of significant reduction in insulin autoantibodies. Now, when we tried to understand what the how these divergent phenotypes were emerging, we went back and thought more deeply about the receptor structures. Now, it should be noted that this is a more complicated receptor complex system than I originally described to you, in that IL-17RA is actually a coceptor for a variety of other receptors as well, and required for their function. One thing that emerged very quickly, though, when we looked at the immune system was the extensive expression of IL-17RA amongst a variety of different immune cells, including T and B cells, and the extremely limited expression of IL-17RC within the immune system. And we confirmed those results through sorting of immune subsets from the NOD spleen, and showed that, in fact, IL-17RA was highly expressed while IL-17RC was missing from those cells. We showed, in this experiment here, we measured the expression of IL-17RA in a variety of different organs, particularly relevant to diabetes, the pancreatic lymph node and islets from NOD mice, and showed a very clear upregulation of IL-17RA in CD4 T cells, CD8 T cells, and B cells, specifically within the islets of the of NOD mice. I'll go through this slide here just to explain what this is. So for a non-mouse immunologist, this is a slightly complicated experiment. Well, what we've done here is taken islet antigen-specific CD8 T cells, labelled them with CFSC, injected them into the mouse, come back 5 days later and isolated a variety of different organs. So these are our transferred cells here with IL-17RA expression on the y-axis. After 5 days, we expect to see no proliferation in this organ, as there's no antigen to respond to. However, when we look in the pancreatic lymph node, we see this dilution of the dye across to the left-hand side, indicating that these cells have become activated and divided. And then finally, in the islets, we see this highly divided population here. What we can see, though, is this incremental increase in expression of IL-17RA as we move from the activated population in the pancreatic lymph node and then into the islets, suggesting a location-dependent function of this receptor, location-dependent upregulation. And we see very similar effects when we use BDC CD4 T cells, islet-specific CD4 T cells. In terms of trying to understand the mechanism of what's going on here, we're continuing to analyse this, but a few things have clearly popped out. A lack of KLRG1 positive CD8 T cells in the spleen. This is is indicative of a loss of important effector lineages in the CD8 population, and in addition, some evidence of reduced numbers of IGRP-specific total CD8 T cells as well. And finally, we've also identified some changes in the B cell compartment, which may be consistent with changes in antigen presentation, particularly a reduced expression of MHC class 1 and class 2 in the knockout mice. Yes, so the final summary and conclusions. I've shown you there that IL-17F has similar pathogenic functions to IL-17A in our in vitro models that we've used, that we've generated these IL-17RA and RC knockout mice, but they actually have quite divergent functions, which was not what we expected to see. Upon our investigation, we can see actually that IL-17RA is widely expressed in the immune system and really does seem to be upregulated in the specific location of the islets during the immune response, suggesting important functions within that context. And I think those pieces of data actually provide new insight into the roles of these molecules in NOD mice and type 1 diabetes. And finally, I'd just like to thank the people who did most of this work. So the IL-17RA work was mostly Johnny's master's degree, and Tara did most of the islet work in the first half of the talk. And of course, thank you to JDRF and Diabetes Australia for funding.